Good afternoon. Welcome back. Hope everyone had a good lunch and a nice break. My name is Nadine Shaheen. I'm an associate professor of orthopedic surgery in joint appointment in orthopedic surgery and biomedical engineering. And it was not that long ago that actually I was in the same seat as many of you here when I was a applicant to the graduate program here. So I'm very familiar with the kind of experiences you're all going through and the excitement and perhaps anxiety that's going through your mind trying to figure out what is the right opportunity and the right fit for you, for your research and your graduate training. So welcome. I also have the pleasure of introducing our speakers and moderating this session on orthopedics and biomechanics. And I want to say that actually this is one of the core components from a topic area for the Department of Biomedical Engineering. We started out as a department historically with our founding chair, Dr. Van Mao, who really instituted orthopedic research and biomechanics research here at Columbia in a strong way that has had long-lasting impact. And so a lot of the people that you'll see and hear from tonight have some touch point with these experiences and history. So our first speaker is Dr. Clark Hung, who's a professor of biomedical engineering and orthopedic surgery, and he's also the vice chair for the Department of Biomedical Engineering. Welcome, Dr. Hung.
All right, so I wanted to thank the organizers for having me present today, and thankfully, I'm not going to talk about science today, but just more about the education and some of the major grants that we have at the university. But I just wanted to echo what George Theodotis was saying this morning, where historically there's been a lot of connections between the engineering school and the medical center, and this really clearly is one of those historically based ones where we really can do some amazing things with really a critical mass of investigators. So before I begin, I just wanted to say that we just got back from Phoenix. Some of our students still aren't back yet, I don't think. But the idea is we went to the Orthopedic Research Society meeting, and this just shows you, I think it's like 40 people, maybe 6 of the PIs were there. But the training at Columbia really has been generational, including many of the folks who are faculty here for decades and longer. And the idea is that not only do we have our trainees currently who are in some of these pictures, but actually sort of trainees of trainees, so actually grandchildren and great-great-grandchildren. So it's one of those things where I think that the legacy of Columbia, at least in the musculoskeletal field, is really sort of a special one. And I think that it's like one of those things, like once you're in it, it's hard to get out. So in terms of being able to sort of excel in the area, I mean, there's been a lot of commitment by the faculty, both to training students, but in addition also to trying to get sort of large grants where we can all work together. So I'm going to take the opportunity to talk a little bit about training first and then some of our larger projects. So the Integrated Musculoskeletal Training Program, actually Nadine is one of the co-directors of the program with Ed Guo, really brings together multiple disciplines at the university, including within engineering, some of the clinical and basic science departments at the medical center. And the idea is really with the appreciation and predicated upon the idea that we understand that moving forward, the next generation of researchers really have to have this sort of mixed background, the ability to communicate with different disciplines and to be effective working in teams. So if you look at the required training elements, you can see that basically co-advising is required of engineer, biologist, and clinician. I would say that probably this has been ongoing for a long time now, but it's been sort of codified or more formalized in this training grant. In addition, with the importance of actually understanding the clinical problem, there's a clinical preceptorship, and the idea is that to really be able to identify, lastly, the important gaps or unmet clinical needs, you really need to understand the clinical problem. So all these elements kind of fall in together to really hopefully train the next generation of impactful researchers. This slide that I borrowed from Nadine basically shows you the infrastructure of the training program builds from many internal people within the institution from the different departments that were listed previously. We're able to build on some staff from a number of departments as well. And the idea is that all these things work together in harmony where students really get sort of a well-rounded image or practice of basically musculoskeletal research. In terms of the training programs, not surprising for NIH training grants, there's citizenship or permanent residence requirements. having a bachelor's degree from one of the departments that's listed here, with the idea that in this particular program, students sort of are in a pipeline coming in their first year. So you guys, some of you may be selected to become eventual potential participants in the training grant, but you actually join officially in the second and third year of your training program. You have to have an advisor who's actually within the training faculty of the program. And again, which makes sense, your thesis has to be related to the musculoskeletal field. In terms of application, there's a rubric here. There's a number of different components that feed into the selection of students. I think that, again, the idea is to try to look forward to see which individuals we feel like will have an impact in the field, that will be synergistic, and with the idea that, again, with the pipeline of students coming in their first year, you can see here there's a couple students already appointed. They'll be on for their second and third year roll off as new students are coming into the program. So the last slide for this particular section, again, just kind of articulates basically how the year 1, 2, and 3 kind of work together, where year 1, you're sort of not officially in the actual training aspects per se in terms of year 2 and 3, but you're basically being funded likely by the department for the first year, being trained by one of the faculty within the program, and then you sort of are selected, if you're competitive, to be able to join the program and continue on. So with that in mind, this sort of is the first part of my presentation. And so the idea, again, the same faculty who make this teaching environment really something special are also those who are involved in trying to bring together innovation in orthopedic research as well. So George also mentioned this morning about ARPA-H. This is a sort of a parallel organization to the NIH under the HSS, the Human Health Services. And happy to say that we were actually the first ARPA-H grant that as a prime organization for Columbia University, and also the first program within ARPA-H that was actually funded, and it was basically based on osteoarthritis. So ARPA-H, we were actually under the NITRO program, the acronym is there, with the idea that this just over $39 million grant is in multiple phases. We're in the middle, we're 2/3 of the way through year 1 in phase 1, which is sort of the R&D phase, I would say. But what's unique about this particular project is that not only is it R&D, but as you'll see in a couple of slides, there's really sort of like the aspect of bringing things to clinical trials really in 5 years. So I have to say, in the words of Nadine, it's really one of those things where it's pretty unprecedented, where we can have the utilization of all the faculty in this particular research area all working in the same problem at the same time. And it's one of those things that are afforded by the big price tag that's here, and things are really moving along well. I just wanted to show here the kickoff that was in Oklahoma in beginning of April. There's 5 teams, so we're one of the 5 teams selected for this program, and it really, is really interesting because, again, what you'll see is not only will we have to bring in a large animal preclinical component, but then actually get into phase 1 clinical trials, but there's also regulatory as well as commercialization. So the problem for us in terms of articular cartilage, which is the specialized connective tissue on our joints, is load-bearing and weight-bearing. And the idea is that when you have articular cartilage that gets damaged or we get old from wear and tear, you can get arthritis and lesions over much of the articulating surface. And when something like this happens, pretty much the gold standard for repair is basically the metal and plastic knee replacement. And so even though these knee replacements work really well and is the gold standard, it's actually still an unmet clinical need, with the idea that there can be complications associated with getting total knee replacements, but the challenge is that they last maybe 15 to 20 years. So it sounds pretty good for someone who's probably in their later stages of life, but for individuals like your age, you'll likely have to go through multiple knee replacements with more challenging levels of success. And this is something we want to try to avoid. And additionally, the number of individuals having to get total knee replacements and also revision surgeries basically is something where it's really growing exponentially as well. So in order to be able to address this, ARPA-H basically wants us to create a living knee replacement, something that can actually change and grow with the patient and be hopefully the last knee replacement you actually need. We have an autologous and allogenic version, patient-specific, needs to form bone, cartilage, and be able to function, and critically, no permanent fixation. So for us to do this, we've been able to come up with tasks from a number of our different researchers, focusing at different aspects of our total knee replacement. Also, we have some teams to look at the animal studies, looking at patient recruitment as well as clinical trials engagement, regulatory, and then commercialization. So in order for us to do this, again, this is a real clinical problem. This is from Roshan Shah, who's the primary physician on our project. It's something that we think will be the next revolution in total joint replacements. We have basically, our team is primarily at Columbia. We have some outside commercial partners in New York City as well. And then our clinical as well as preclinical partners are at Missouri. Jimmy Cook is the head there in Columbia, Missouri. Again, as we've alluded to previously, really NovaJoint really stems from the historical strengths in musculoskeletal biomechanics and then emergent strengths in biomaterials and tissue engineering. And so these are just some examples of folks here. Jimmy Cook is pictured up here. He's the veterinary orthopedic surgeon. And the idea is that to lead, whatever, 10 or 12 PIs, basically we've been able to sort of build on the historical background and strengths. And this is just a smattering of the kind of research that the team members have brought to the table and actually probably what got us funded, the ability and the promise of being able to do this major ambitious project together. To lead the team, I'm fortunate to be working with Nadine Shaheen, and then we were fortunate to have some new friends and colleagues, Robin and Amnea, who basically round out the team. And so we're trying to right the ship and move things forward through all the trials and tribulations of this program. And then the last couple slides, we can't really do anything in terms of commercialization without Columbia Technology Ventures. Domina Koo is our technology licensing officer, and she's actually an alum from our program as well. We've also hired a business consultant. We have an insurance action plan that's headed in part by an individual at University of Missouri. And then under regulatory, we have a consultant, Laura Silverman, who actually is a grandchild of our program in a sense that she trained with one of our trainees. So it's a small world. It's hard to get away. And with that in mind, things are going pretty well. October, we had our first site visit. So here's some of the Nitro folks with us. We have our second site visit coming up in May, I believe. So hopefully that will go out as well. And with that, I just wanted to thank you guys all for your attention. If there's questions later on about the T32, basically Nadine can answer that, and then ARPA-H, I'm happy to answer it. So thanks very much.
Thank you so much. So our second presenter is Dr. Steve Thimopoulos, the Carroll Professor of Biomechanics and Orthopedic Surgery, Biomedical Engineering, and Vice Chair for Basic Research in the Department of Biomedical Engineering. Welcome.
Thanks, Nadine. So since we're giving some historical context, actually my first exposure in orthopedic research was as an undergraduate here in the mid-1990s in Van Mouw's lab. So I took Professor Teshin's biomechanics class and never looked back. So it took me 20 years to come back to Columbia. I've been back for about 10 years and actually in charge of the space where I first got exposure to orthopedic research, so full circle. Okay, so I'm going to talk science. I'm going to talk about some of the research in my laboratory. And this is the clinical rationale, kind of the basis for the problem we are really focused in trying to solve. And it comes down to this issue of trying to reconnect a connective tissue like tendon or ligament back to bone. In the rotator cuff, for example, hundreds of thousands of these repairs each year, with a pretty alarmingly high failure rate. So the surgical approaches right now are not nearly good enough. And so this motivates us to try to understand why it doesn't heal well, what we might be able to do to improve these outcomes. The basic problem is fundamentally a mechanics problem. You're trying to connect a rope to a piece of cement, tendon to bone, two materials that are very, very different, structurally, mechanically, compositionally. You have tendon, which is relatively extensible, bone, relatively brittle. You connect two materials that are this different, you're always going to end up with stress concentrations at their interface. Now, of course, the body has solved this problem through a number of mechanisms that I'll talk about, but ultimately the problem is that the healing process does not recapitulate the structure that works so well when it's healthy. This is the strategy we take in my laboratory. There's two essential basic building blocks that we work from. The first is to try to define the design criteria. What does the ultimate tissue need to look like? And we do a lot of biomechanics, structure function types of studies to try to connect the structural organization, the compositional makeup of this attachment structure and relate it to the biomechanics. So this is a design criteria that we're trying to achieve. The second big area is to define the roadmap. So how can we get the cells that we have available, the biology, to rebuild this really elegant structure that works so well when it's healthy? And we look to developmental biology to get those lessons. We put those two things together with regenerative medicine strategies to come up with solutions. So this might involve scaffolds that recapitulate some of the design criteria from the natural system, progenitor cells, biophysical cues, bringing them together in animal models, typically to test out regenerative translational strategies for repair. And this is what we're trying to achieve. The enthesis is the anatomic term for the attachment of tendon to bone. This is this really transitional structure that goes between tendon and bone. And you can see here that there's a gradient transition from highly aligned collagen fibers on one side, on tendon, to this highly mineralized stiff tissue bone on the other end. And in the middle, you have this gradual transition across this fibrocartilaginous structure that includes a mineralization gradient as you go from tendon to bone. For the biomechanics studies that we do, we try to think about this in a multi-scale way. So everything from the tissue level fibrous architecture that dictates the mechanics of the rotator cuff tendons, for example, to the mineral architecture, these gradients in mineral that are on the micrometer length scale, all the way down to how mineral accumulates on individual collagen fibrils. When it does fail, what happens if you take a tendon and pull on it in the laboratory? It's going to fail and look like the image post-failure that's shown here on the right in these micro-CT images. And what you'll notice is that this big chunk of bone typically gets pulled off. So even though you would intuitively think that the weakest point might be the attachment site where you'd have stress concentrations, in fact, that transitional zone is so tough and resilient that it actually can pull out a piece of bone typically when it's healthy. An example of the types of studies we do in that biomechanics set of studies. Here's one example where a graduate student looked at the effect of the orientation of the fibers as they insert into the bone, modeling that behavior, and really identifying that there's this balance between toughness and strength that's achieved in the rotator cuff tendons to get an optimal scenario to prevent rupture of that attachment. Okay, so if we look at the healing process, here's the current state of the art. And this is what, if you tear your rotator cuff, you go to the shoulder surgeon, this is what they would do. A typical repairer might have a few suture anchors that anchor those sutures into the bone, and you grasp the tendon with this cross stitch like this and hope for the best. And the problem with this is that, as you can imagine, all of the stress has to be transferred across 2 or 4 transfer points. You don't have a good distribution of stresses, and this fails quite frequently. A key reason why it fails can be seen if you look at the histology of the healing process. So on the left here, you have a healthy attachment, tendon on the top, bone on the bottom. The images on the bottom are polarized light microscopy images which highlight the orientation of the fiber. So you can see in the healthy attachment structure this really beautiful connection, continuous fibers that cross all the way from the unmineralized tendon into the mineralized bone. And you contrast that with the healing situation on the right, and you can see that there's a very abrupt interface with the mineralized bone on the bottom and this disorganized scar tissue that forms on top. So I think you can imagine that if you pulled on that structure on the right, it would fail pretty easily. How do we define the roadmap to regenerate these structures, get it to build the right thing and not this scar tissue that is so mechanically weak? We look towards biophysical and molecular cues to try to come up with strategies that we can implement in the healing setting. I'll talk a little bit about one area that we've really focused on in the recent years, and that is on this particular cell population that we think has a lot of promise to try to get regeneration to occur. And so what I'm showing here is a mouse model where we're tracing cells that are positive for GLI1. GLI1 is just the factor that is indicating that the cells are activated for a particular pathway, for Hedgehog signaling. And what you see here is that early after birth in this mouse model, you have a few green cells. These are the GLI1 positive cells. If you track the lineage of these cells, or all of the daughter cells, and you go out to an adult skeletally mature time point, 56 days after birth, you see that it lights up across the entire attachment. So that interface, that enthesis between tendon and bone, all of the cells that are there are lineage traced from this original stem cell population that was positive for GLI1. Okay, so that this means that these are the cells that build the entire structure. And if you ablate all of these cells early on, you don't get the formation of a functional attachment structure. So wild type normal is on the left. You see that red staining shows the fibrocartilage that forms. If you kill those cells early on, you don't form that fibrocartilage. Very little red staining. So how can we use this in terms of a regenerative strategy? Well, we took this a step further to try to understand what are these stem cells that are so specific to this attachment structure that build this really nice transitional tissue. If you isolate these cells using flow cytometry, so you see the GLI1 positive cells, those blue cells that are shown on the upper left there, you sort those out, and then you further sort those into cells that have markers for stem cells, Ly6A and CD44 in this case. And what you'll see if you do a standard colony formation assay, which is one indicator of stemness, stem cell capacity, these GLI1 stem cells are the most potent in terms of forming these colonies. And even more specifically, if you put them in culture and hit them with appropriate factors that might promote multilineage differentiation, you'll see that they don't have the capacity to form fat cells. So adipogenic media, mesenchymal stem cell, standard bone marrow MSC, can form this positive staining for adipogenesis. These GLI1 stem cells are already committed beyond that, so they're not able to form fat, but they are able to form bone, as shown here, and very potently able to form cartilage. So they have pluripotent capacity, but they are already tracking towards this enthesis phenotype. We looked even deeper at these cells. This is a single-cell RNA-seq experiment where we traced those GLI1 positive cells, and we looked at their transcriptomes across the whole lineage from their stem cell all the way down to a mineralized chondrocyte, mineralized fibrocartilage. And they follow this really pretty straight and continuous path of differentiation into enthesoblasts, chondrocytes, and then mineralized. Applying this in the real world, let's say, for something practical and useful translationally, we looked at how healing processes occur in the mouse model again. And so what this shows is that same lineage tracing on the left, an uninjured enthesis. And on the right, if you injure early on, postnatally, when there's a lot of these GLI1 positive cells, the entire enthesis effectively regenerates. You can see that structure on the right. The asterisk shows you where the injury originally was. It's pretty hard to find. And this is what it looks like if you do the injury in the adult, when there aren't many of these stem cells left. You have this scar tissue, a little bit remnant of the enthesis, shown here on the right in the green cells, but on top of that is just a mess of a scar. Now, if we actually create a therapeutic here, the idea is to take these Gli1 cells and deliver them to the injury site. So what this shows is what happens if you do that. And you can see bone volume, for example. The white boxes outline where the injury was. You have a significant increase in bone volume with the treatment of these Gli1 stem cells. Similarly, histologically, histologic score of 0 is normal. So you can see over time, post-injury, 7 days, 14 days, and 28 days, you see this improvement, but a much better healing process with the stem cell therapy. And most importantly, functionally, you have this significant increase in all of the metrics of strength and stiffness, toughness that you would hope to achieve in a better healing response with these Gli1 stem cells. Okay, so to wrap up, the approach in my laboratory is to really take the basic science, biomechanics, understand what the design criteria is to effectively get load transfer between tendon and bone, look to developmental biology to come up with strategies and stem cells and growth factors that might be applicable, and then apply these lessons in regenerative strategies in animal models. So I'll end there. This is a picture of the Carroll Laboratories. We are 3 PIs, Dr. Shaheen, you can see there, Dr. Alice Wong as well. And this is most of our group up at the medical center, the laboratories for orthopedic surgery, where we focus on studies like similar to the ones I talked about today. All right, thank you.
Thank you. Thank you, Dr. Thermopoulos. Our next speaker is Dr. Andrew Lane, the Percy and Vida Hudson Professor of Biomedical Engineering and Radiology, and is on the executive leadership of the Biomechanics program, which we're going to learn all about now. Welcome.
Thank you, Nadine. So historically, since everyone's talking a little bit of history, this talk may seem a bit out of place, but this is kind of the old guy session, I think, because looking back when Clark, as Nadine mentioned, biomechanics was the strongest and the first pillar of strength as we built the department. And I believe that both Clark and Gerard Ateshi, and I think the department wouldn't exist without their work. I remember a famous 3 ropes that I think Clark developed that slide that moved us forward in getting grants from Whitaker and onward down the road. So it's really a quite strong group we have in this session. Okay, with that in mind, I thought some of you are here because you probably want to have an impact in saving lives within your career in the short term. And Columbia offers a wonderful program called Biomechanics. It was originally called the Wallace Coulter Translational Program back in 2012. And that program transitioned, but it's been more or less self-supporting ever since. So I thought I'd give you a little bit of a snapshot of what this program looks like. And so it is an opportunity for faculty and students to leverage the university's resources and the Presbyterian Hospital, which we're associated with, as well as the business school and School of Public Health. And the main focus is, again, to address a clinically unmet need that can only be solved by teaming together engineers with clinicians. And so, of course, the overarching goal, as I mentioned, is to improve healthcare in the population, reducing healthcare costs, and to translate the discoveries that you all are making in your laboratories into commercialized products such that they can hit the market and actually begin to save lives. And of course, this is the famous diagram of the Valley of Death. And most of us spend our time on the far left-hand side in blue. We do a lot of research and day in and day out, and we publish our papers, and that's very nice. However, it's really a shame that some of that technology never gets transitioned into a commercialized product to benefit humanity. And there are opportunities along the way, such as SBIRs and STTR type of programs that could bridge this gap, but it's a difficult one. And this is where the Biomechanics program is designed to fill. But this requires a combination of education, specifically commercialization. And most of the professors, at least I didn't know much about commercialization. I came from a pure academic background, but over the last, I think the programs are running now for over 12 years. I think it started in 2012. And so I've also learned a lot along the way. But it's really a combination of having highly motivated people like yourself and students, and having the knowledge of the business, marketing, who pays for the device and so on, as well as developing the underlying technology. So it requires all three of those to build the program. And at Columbia, we have that. We have an excellent pool of clinicians, engineers, and on the far right-hand side, you'll see you all there, the students and postdocs, as well as we have resources within Columbia for advising you on IP, and we have external consultants to talk about FDA approvals and analysis and mentors. Also, so we have a very good culture within here, and the areas historically where healthcare innovation has proven successful including the delivery of healthcare, understanding the workflow of a clinician, and having a device or a technology that could expedite that or reduce the cost. But historically, the Biomechanics program is focused on technology that facilitates the movement of a procedure from the inpatient to the outpatient setting, or from a traditionally brick-and-mortar visit to a virtual type of setting through monitoring, for example. And this includes digital health products that can address critical care. And this touches upon many departments within Columbia in many areas, including surgery, therapeutics. I'll talk about a little bit more later. Rehabilitation and robotics, psychiatry, imaging is a strong area, immunology, of course, and cardiology. But a key resource that Columbia offers is a makerspace. So when you're developing a device, for example, you may want to work with a prototype. And so just know that we have incredible resources both on this campus and on the medical school campus to fabricate and to develop such devices. And so as I mentioned, historically, this program has focused on primarily devices, but it has more recently migrated over to therapeutics. And there's another accelerator at Columbia called T-REX, which stands for a Therapeutic Accelerator. And there we often partner with them to sponsor jointly some ideas that may look promising in terms of IP and marketing. And this includes cancer through the Cancer Center, COPD, pulmonary, lung diseases, and cardiovascular, as well as other areas. But this space is very complex, and it can drain you. And the yellow arrow across it requires a lot of time and energy. And so you have to be really committed, strong to make it through this area. You'd have to identify licensing, FDA regulations. It's a very complex space. And so don't get discouraged, though. I think we provide you with the infrastructure to guide you through this space. And if you look at our website, you'll see many successful programs and products that in fact have come to market or have been licensed or have received venture capital funding. So it all starts with a education, and we have a fantastic course. It's called the Lab to Market course, or a boot camp course, and I'll talk more about that in the next slide. And also we have business partners, the School of Business, their faculty and students are involved with the program. We also provide you with an active program management within what's called an oversight committee. And I believe personally the oversight committee is really the special sauce that makes this program successful. These are experts within various fields of medical engineering that, they help us. They don't get paid much, actually. We pay for travel every now and then, but for the most part, they're just giving back they've made their fortunes, they've marketed their products, and now it's time for them to give back. So they really, and they tell of their successes, and more importantly, they tell of their failures, what didn't work. And so it's that knowledge, that insight, that really makes the difference. Because if you look at the size of the funding, there's not much money on the table here. They're about $100K, which is really small change by comparison. But the insight, the value is provided by the Oversight Committee. So let's take a look at the the Lab to Market course. And there's a required section that looks at, oh, thank you so much. Yeah. That looks at the validating the business case in terms of the value proposition and what, who's going to pay for it. You have to have a product, you have to have a strategy, there has to be a insurance code or something existent that'll allow it to be marketed, as well as what we focus on is how do you de-risk your idea such that it is very tantalizing to a venture capital person to further invest in it. And that's done by identifying what's called a killer experiment. So the killer experiment is such that if you succeed in this experiment, it'll either kill the idea and you realize that there's no future here, I can go on and work on other types of problems. Or if it fails, that means you're really on your way to success. And there's the other part of the course, shown on the right-hand side, is, for the most part, consists of outside speakers that come in with specific knowledge about various markets. This is the syllabus, so it's run by Megan. Is Megan here? I'm not sure, but Megan Egan is one of the people that run the course along with Dennis Purcell from the business school, and there's also help from the IT office as well. So to be eligible for this program, you need to interact with a faculty member and bring along a clinician or an engineer, basic scientist type. But the important point is this is a wonderful opportunity for graduate students and postdoc trainees to act as project leaders. Now, in my experience, I've been involved with this program now since 2012, and there's been no professor that has left to start a company from Columbia. It's always the students. They're young. What do they get to lose? At that point, so they go out and they really take the leap forward, and so that's really an opportunity for you. And of course, licensing technology is a key aspect. You have to work with the licensing technology office here at Columbia to check out the space that you're not working in an area that's perhaps too crowded or fuzzy. And so we have people that will help guide you through that process. So here's sort of a timeline of what the cycle looks like, starting on the far left-hand side. We accept proposals in early November, and we usually get about 30 or so. I think this year we had 36 or so. And then I strongly advise that you take the boot camp course in parallel if you're inclined to do this. In parallel, then you'll you'll have to present a full proposal if you're selected. And then there's something called a pitch day, and we get down to about 10 pitches during that process. And we finally find up funding about 3 to 5 types of proposals. This is a picture of the leadership team. Megan Pinach recently left us, but Megan Hina now is in charge. Anil is the clinical leadership side. And as I mentioned, Dennis Purcell from the business school runs it. And Paul Scheid and I, who's the chair of the department, are the PIs on the biomedical engineering side. I think I'm running out of time here, so let me just speed up. It's been a wonderful program. We've gotten very good feedback both from students and faculty. We get sort of a 10%, if you look at from a business, a sustainability point of view, we get about 10% return on the investment. Better than the stock market, so that's pretty good. In terms of, if you look at the total amount of revenue, over the last 12 years or so, we funded about $5.8 million or so, and this follow-on funding, though, is really what I think makes the deans happy, is because even though we bring together clinicians and engineers together, and they may not get funding from us, but what they discover is that they need to go back and do some more research, something more fundamental, and they'll go out and apply for an R01, and they're likely to get that. So it's really a win-win for both the school and the program. So that's what follow-on funding means. Again, I'm running out of time here, but we have people from the Northeast and around the country that serve on our evaluation team. There are other examples of these programs, including at Penn Tech Health and at Harvard and at Stanford, which is one of the first. But we were also a Coulter school, and we've managed to continue the program beyond Coulter funding. Long term, our dream is to have this program sustainable from a foundation of some kind. We're not quite there yet, but perhaps someday. And just sort of quickly, this lesson learns over the prior experience. You need to have clinical pull, sort of addressing the needs or problems experienced by the patients and clinicians is more powerful than just pushing a novel technology alone. And that in terms of investment, early connections outside of the academic sphere are really critical. And building relationships within your team, that's also very important. The fact that we provide an oversight of the program to keep you going and address any problems as they emerge is also a success. So on that note, I'll thank you for your attention.
Okay, and our last speaker in this session is Dr. Gerard Ateshin, the Andrew Walls Professor of Mechanical Engineering and Biomedical Engineering. Welcome.
Thank you. So I'm going to give a basic science talk. So I'm just going to jump in. I have exactly one message I want to convey. So I do a lot of work in cartilage, and it turns out that cartilage wear is caused by fatigue failure. And this may mean nothing to you, but historically, people look at cartilage. So these are different joints. It's not the same joint over time. Obviously, we can't do that. But you can see that cartilage is healthy on the left side, less healthy in the middle side, and on the right side, this is full-blown osteoarthritis. So for a long time, meaning as far back as the beginning of the 20th century, people simply assumed that wear was caused by friction. That was like the prevailing assumption. But the fact is that most of our joints undergo a lot of motion and reciprocating loads. So therefore, you could say, oh yeah, reciprocating friction is the cause for the wear. But how much wear is really happening? So historically, one measures the friction coefficient as the ratio of the frictional force to the normal force. That's the formula on the right. What you see on the left is a book chapter that I wrote with my advisor when I was still a graduate student. Had 240 references going back to the beginning of the 20th century. So I knew the literature really well when I started this work. And everyone was doing friction. Very few people were looking at wear, even though osteoarthritis is really a wear problem. So basically, in 2009, with one of my students, we did experiments where we measured the friction coefficient in arthritic human tissue. On the top left, you can see that it's less arthritic. We can get perfectly healthy joints, and that's India ink that we use as a way to figure it out. And at the bottom left, you see highly arthritic. And if you compare the friction coefficient, which is the graph on the right, as a function of time, on the vertical axis is the friction coefficient, you see how small it is. It's 3% of the applied load. So if I'm standing here and I'm applying 2 body weights on my joints, the friction coefficient is only 0.03, 3% of 2 body weight. It's tiny, and it's even less than arthritic cartilage. So the idea that wear is progressing to increase friction is ludicrous. You would think everyone would have read this paper in 2009 and said, okay, we give up on the idea of friction. But no, they didn't. So we have to, get more funding and show people things. So this is Krista Durney in 2020. Krista is a BME PhD student at the time, became a faculty later. But anyway, so ran this experiment where you have a glass lens sliding on a piece of cartilage from immature bovine cartilage. And I'm going to skip this slide, and I'm going to show you the next one, actually, and I'll come back, and caused wear. And the wear that you see on the left is a blister that's formed, and it's away from the edges, so it wasn't an edge effect. And on the right, you can see polarized light microscopy cross-sections that she got from multiple samples that show you that there's delamination. So cartilage wear is not abrasive. It's not like using sandpaper on wood and you see particulates of wood coming off. It's not abrasive wear. It's delamination. If I had a laser pointer, maybe I do, who knows? No. It's the red light. There we go. You see? That's a blister. It's separated. That's what we call delamination. Here, right there. Okay, good. Sorry. Okay, so back to the previous slide. And yet Krista found that actually, for some of the cases, the friction coefficient went up, and for some of the cases, it didn't go up. There we go. So the friction coefficient did not correlate with the wear, and it remained low in any case, and even when it went up, it went back down eventually. So it didn't seem to matter. So it's not friction. In her experiments, she had also done cartilage on cartilage, and this is what was interesting. She found that on this side, which is cartilage also, it's from the condyle, it never wore. The blister only appeared on the strip. At the time, we didn't make a big deal about it, but then in hindsight, we were like, wait a minute, what's the difference between the top and the bottom? The bottom is seeing reciprocating load. If you fixate at one location, the load goes up and down, whereas the top sees the load constantly. It's a constant load. The top is not reciprocating, the bottom is reciprocating. That's fatigue failure. So that means that the joints were actually undergoing fatigue failure. But we figured that logic is not going to sell. You got to give more direct evidence to people who read papers, because scientists are like children. They're no different than you and me. So you got to explain things. So we did an experiment where we made sure that the reciprocating glass slide was causing the load to go up and down on the cartilage, or the glass slide was so big compared to the cartilage that the load was constant. And sure enough, this is a study by Courtney Pearson. Here's Courtney. And basically what she did is she showed that if you actually move the lens, you're articulating back and forth, you're actually causing the load to increase and decrease at this location. Whereas for this configuration, which we call the stationary contact area, the contact area stays the same, the load stays constant. And this is what she found. She found that for 8 out of the 8 samples that were loaded in the reciprocating in a migrating contact area, they all delaminated. None of the samples that were in this configuration delaminated. This is an example of those that delaminated and didn't, right? So that's direct evidence that it's the reciprocating load. This is experimental evidence. I'm not bringing any theory, although I love theory. And this is the friction coefficient in the case of delamination. This is the friction coefficient for the stationary contact area. It rises over time, and it's higher. So the friction coefficient is higher in the samples that didn't damage. You understand? So it cannot be friction. Okay, so I think we finally convinced the literature, on this point. And that's it. That's the message that I wanted to convey. Thank you.
There is no doubt about it. We're going to ask our speakers to come up here for a panel discussion and some questions. And I'm going to pass, while they take their seat, we're going to pass the microphone around to the audience. Yes? Oh, Cameron?
Yeah, you can use the podium mic.
Okay, thank you.
Thank you all so much. Thank you all so much for your talks.
My question is specifically for Dr. Tomopoulos. Thinking about the translational aspect of these GLI1 positive cells, have you had issues with cell viability post-transplantation? And then related to that, what are the other challenges that you foresee for the clinical translation and commercialization of a GLI1 positive cell-based therapeutic? And do you have ideas for how to overcome those challenges?
Yeah, great question. There's a lot there. So the basic science work, entering into translational arena is what I showed, mouse model, not really a relevant type of injury that recapitulates rotator cuff, et cetera, et cetera. That proof of concept shows that if you put the cells there, which you don't have many of these cells left in adulthood, if you put them there, they have the capacity to regenerate that enthesis and start to build that structure. The application of that proof of concept has a lot of steps left to it. First and foremost is getting the cells, as you're implying, there and keeping them there and maintaining their phenotype in the presence of, say, all of the inflammatory factors that are around during wound healing, et cetera. So we're working on scaffolding and various cell approaches that can achieve that. Source of cells is also important. In that mouse model, we can get actual, original GLI1 positive stem cells. In reality, you're going to have to use autologous, bone marrow stem cells, and push them into that phenotype. And then the final bit, the actual implementation and commercialization. The Biomechanics talk kind of showed you that path, and that valley of death is very real. So a biologic like this, a cell therapy, has a lot of hurdles, and a lot more than a device, for example. So quite a ways to go. We're in the small animal, maybe into, say, rabbit-level translational studies now, and it'll be years before, let's say, hopefully, a clinical trial.
Thank you to all the panelists for your amazing talks. My question is specifically for Professor Lane. I just had a question about the Biomechanics program, which is such an exciting opportunity for young scientists and people who are looking to make an impact. I guess what's interesting is postdocs as well as graduate students are allowed to be in this program, so there's a lot of different types of data that's coming in. So there can be a really great idea with maybe a little less data than the others. So I'm just wondering, when looking for projects, devices, or therapeutics, what kind of data do you need before you submit a project for, a program like this, more preclinical animal data, or, I was just wondering how the projects are assessed.
Yeah, that's a great question. Is this on? Can you hear me? Okay. So the Biomechanics program is not for basic research. So we expect that at that stage you would have got your NIH grant and you would have had some preliminary data already. Again, the amount of funding that's available from the program is simply to catalyze the transition across this valley of death. And so it will not, that said, however, we have funded animal studies to generate more convincing data that was needed for example, a killer experiment. So it really depends on the application, but for the most part, Biomechanics is not a place to go for funding of fundamentals of the science and research, but more, if you apply, you'll get a message saying you're too early, come back later when you have some more convincing data. So that's yeah.
Dr. Lane, I have a related follow-up question. So are the projects already have a patent or IP application that has been filed with the Columbia IP office, or does that happen in the process of being in the program?
Usually you would talk to the intellectual property to discuss anything that when you apply to the program, it's confidential through a secure website. And I think Oprah's back there. She's probably the best person to answer that question, but certainly we advise you not to publish if you have a great idea and you're convinced this might be commercializable, not to publish it, hold back, and file the patent first. And once that patent is secure, then you can publish it and talk about it. So that's really important because sometimes you you can lose the opportunity by publishing ahead. Yeah. Yeah.
Can you pass the microphone?
Oh, yeah. Here.
Representing the Tech Transfer Office, CTV, we are happy and interested in talking with all of you, any of you, early. And so don't wait until it is clear to you that you have an invention. Come talk to us. We're happy to brainstorm with you and just sit down and develop a strategy with you for your research.
Question in the middle.
For the GLI1 positive cell treatments, I know that there is some research that there are people who have a genetic predisposition to rotator cuff injuries, and I'm wondering if this treatment would work on those people. And also, is there a way to enhance the capability of the cell, like beyond what already exists in nature, to potentially accelerate the regenerative process?
Yeah, so rotator cuff disease, it's multifactorial, and there is some genetic predisposition, but for the most part, it's an age-related condition where you build up chronic degeneration, and then some event leads to tearing of that cuff. And so what you end up with is a fairly degenerated cuff without many of these cells left by that point, usually older, past 65-year-old typical patient. And so I think that is the setup. That's what the surgeons presented with. And so it's very attractive to come up with a biologic that its origins are in the development of biology, those cells that were there that built it to begin with. The strategy for achieving that is going to be hard to implement, right? Because you're obviously not going to get those original perinatal cells that have all the capacity. You're going to have to turn in adult cells, turn them into that phenotype. Ramping up the behavior, that's a tough one. I think the first step is just to get a cell source that you can push into this phenotype and use that. That's ambitious to begin with. There is some capacity to just turn on the signal, which would be a lot easier. We did a study in a rat model where we just ramped up the signaling without providing the cells, just a systemic drug, and that had some beneficial effect. It didn't regenerate the attachment, but it did improve things. So clinically, the strategy to take is probably incremental and maybe something that will improve, but maybe not regenerate, might be the first step, and then ultimately get to the point where we put in these cells in a proper scaffold and really guide the whole thing.
Hi there. Thank you for your talks today. I had a question about like the living knee replacement and the enthesis. Is there a consideration for sex specificity when creating these regenerative strategies? Is that something you consider at all? And if so, is it at the beginning of the process, or is that something that you would tackle further down the line?
In terms of the Arbor H project, the aspect of sex was actually one of the things that they had mentioned in the broad agency announcement. And I think that from our perspective, I mean, if you look at conventional knee replacements, I think there have been efforts in some cases to make gender-specific implants. I don't think they've made a particularly major splash in things. So I think that, our approach is sort of a hybrid where there is personalization, but I think that ultimately we need to be looking from a commercial perspective to be able to scale. So at this point, I don't think it's a critical aspect of our design aspect, but I think that it has been something that we've considered and we've discussed even from the in vitro testing side of things, whether we needed to look at that variable. Interestingly, I think we were told not to do it because, you don't have enough money to do that. So I think they kind of just said, get there, get to the next point and move on. We were like, okay, so yeah, thanks.
Hi, thank you all for the talks today. I have a question focused for Dr. Hung. So I'm very impressed by the work you've done on the ARPA-H grant and focusing on a knee joint replacement therapy. As far as looking at osteoarthritis and other maybe more complicated joints, is that something that you're looking at, or is that something that is in a future direction at all?
So actually, that's a great question. I think we're probably being recorded, but I think that, to be honest, if we had our option, probably knee would not have been the one to choose. That was sort of something that was prescribed to us. As Gerard can tell you, basically, of all the joints, it's probably the most complex, and biomechanically as well as just structurally and those kinds of things. And I think that we do think about that on our free time, the other potential applications, I guess I would say. And I think that to some degree we review what we're doing as a team, and definitely, again, we're fortunate to have this, and it really does reflect the nature of the individuals who are at Columbia. But the idea is that it's a really kind of a platform technology potentially. So I think that there may be wins in a number of different places, including possibilities for veterinary medicine, depending on which animals are chosen for the actual project. So even from a commercialization perspective, the question might be, how do you sustain capital and activities to get into the clinic, and very much aligned with the kind of things that Dr. Lane was talking about, Biomechanics, this whole strategy of how to avoid that valley of death once the ARPA-H money sort of kind of ends and tapers out. So thanks for that.
Okay. As we pass the microphone on, I wanted to ask Dr. Ritesh a question. I was wondering, given your findings, if you can comment on the role of joint lubrication and wear, and whether your data suggests that some of these clinical treatments like HA injections have an ability to delay wear or slow it down or anything to that effect.
So most patients who present with symptoms like knee pain have lost their cartilage, so there's really nothing to repair. So unfortunately, then the injection of hyaluronic acid or hyaluronan is not going to help. But we think that surgeons occasionally look into a joint for other reasons. Let's say they're doing debridement of a joint, and they look with arthroscopy and they see that the cartilage seems to be getting softer, maybe it's fibrillated at the surface. Could they repair it? So personally, I'm working with a colleague of mine on cross-linking of the collagen using laser light as a way to strengthen it. And our experimental studies show that indeed you can reduce the wear rate by cross-linking the collagen. So, but there has to be cartilage left, which means you have to be, by chance, noticing. Or, like sometimes surgeons will operate on a patient who has a fracture in a joint, but only on one side. So they do a joint replacement, but only on one side. So you could have maybe the humeral head of the shoulder replaced, but not the glenoid, or vice versa. But then they know from experience that the cartilage on the side that they kept intact is going to wear away faster. So maybe if we could strengthen it with cross-linking of the collagen beforehand, then we might prevent the fast progression of away. So those are the kinds of projects we're working
on. My question is also directed to Dr. Artusian. My background in mechanics is rather rudimentary, so I just wanted to make sure that I am taking away the correct takeaway from this. Is there anything unique to cartilage specifically that gives it this property where the wear is independent of the friction based on the fatigue failure? Or would you expect similar results in a prosthetic or mechanical joint?
I think there are things that are unique to cartilage. So prior to focusing on wear, we spent decades looking at structure-function relationships. So cartilage is made of type 2 collagen, which is fibrillar. It's very dense, but it is very porous. So it has a very high water content. Near the articular surface, it's 90% water, but the pores are on the order of 50 nanometer in dimension. So it has very low permeability. It's like a kitchen sponge, but you would have to apply a lot of force to squeeze out the water, but it has a whole lot more water than a kitchen sponge. It's impossible to synthesize it from scratch. So that's why we rely on cells to regenerate it at this time. So it has unique properties, nothing like metal and plastic or anything like that.
Okay, So thank you so much to all the presenters and to the audience for a very engaging discussion and panel. We're going to take a 10-minute break and then continue on in our program with the genomics session at 3:45. Thank you so much.